So Kristin, I think we're self-introducing, so I'm going to let you come up here and introduce yourself. Obviously, you're Kristin Baldwin.
Okay, thanks, Abe. I'm really thrilled to be here at this. I hate these things. I'm thrilled to be here at this meeting where there's a lot of systems biology because we really need some collaborators on this, as you'll see with my talk. And also to follow Abe because I really think the rat is a fantastic model, and we're not talking about it, but we're making mouse-rat chimeric animals with different brain things. So maybe we can discuss that after the break. I often open my talk with this picture from a sort of an art project called the Humans of New York, where they go take pictures of people and interview them, and you get a sense of our similarity to one another as humans, and yet our incredible diversity. And as we all know, human genome sequencers are turning that beautiful diversity into lists of SNPs, very unromantic. What my lab is trying to do is turn those lists back into something biologically interesting and informative. And as Abe pointed out, the mouse has been a great model organism. I love my mice. Rats are a lot smarter and better. And what's seen here is the human genome overlaid on the mouse genome. And so you can see the white parts are, if I can do this correctly, the white parts are the parts where there's no homologue between mouse and human. And so the differences between humans that control our behavior can often be found in these regions. And actually, we have a paper coming out in Cell studying one of those regions for coronary artery disease that links a bunch of GWAS things together. So we're really quite interested in that. And so when you rearrange these colored parts of the genome, instead of making an animal like this, you make something that's obviously far more sophisticated, intelligent, humorous, such as this. I had to put Trey in the picture. Okay, so the other thing to remember about studying neurologic disease is I like to point out the Stephen Hawking problem, which is if you have a neurologic disease or a psychiatric or intelligence phenotype, you need to study that in the right neuron. If you were to study why Stephen Hawking can conceive of the beginning of the universe looking at his motor neurons, you'd probably get the wrong answer, and vice versa if you wanted to study ALS looking in the hippocampus. And this is important because those of us in the stem cell world have been very limited in our ability to generate neurons of known subtypes. And people coming from the genomic world also often don't know which subtype of neuron is actually affected by a disease, such as in schizophrenia or autism. So this is one of the problems my lab is trying to solve. So you have in your brain an unknown number of subtypes. I like to say, you see a neuron, you can recognize it, and Cahal's tracings of neurons basically are still the way that we best can define a neural subtype. But more recently with single-cell RNA-seq, we're starting to get lists of genes that we think can also divide neurons into subtypes. And I'd like to say this is super exciting, but these don't always overlap. And so what my lab's been trying to do is look at the problem in a different way. And that way is to use transcription factor-based reprogramming to make human cells in a dish and try to understand when we make these cells, what does their transcriptome output look like? And can that tell us something important about disease, subtype identity, and help us even maybe in vivo reprogramming one day? So the first way we could do this is to make iPS cells from fibroblasts using 4 transcription factors, a pulse of that. And then you can differentiate these cells by following developmental paradigms, and it works. But the cells you get out are often immature, and it can take very long to get certain cell subtypes. So they're useful for developmental processes, but perhaps not as useful for aging or diseases that affect a more mature brain. A second way to make neurons was discovered initially by Marius Wernig, where you could actually take fibroblasts and directly induce them into neurons in about 2 weeks with a transient pulse of just 3 transcription factors. And this is a very sort of exciting result and lets you try to make neurons. But when we saw this, we'd been thinking about this, and we thought, that's great, but it really shouldn't work, right? I mean, evolution did not select for us to make fibroblasts directly into neurons at any point in our lives. So why does it work? So we have a little hypothesis that will inform the data I'm going to show you later in the talk, which is that maybe what direct reprogramming does is it sort of reboots the cell into a program that evolution has selected for to be stable. And that may represent maybe the programs that mature cells use to maintain their identities and keep the right genes on and the right genes off. And so my lab has sort of followed this hypothesis in a number of ways because it would predict there are more ways to make a certain cell type than you might think. and that the cells that you get out will be at least something important represented by cell types in your brain. And so when you look at neurons in particular, normally they develop through a neural stem cell precursor. They exit the cell cycle irreversibly. They're born when you're born mostly, and then they become mature and make this whole brain. And what we think we can study using reprogramming is the kinds of patterns of gene expression that are common to all neurons, if such patterns exist, and the cell intrinsic kind of diversity. We do not think we can recapitulate the things your neuron learns from its brain. But these, remember, are the things that RNA-seq, single-cell RNA-seq, is reading out in the transcriptome. So if you try to match a neuron in a dish to a neuron in a brain, these things should not match. Okay. So the way that people, after Marius published this paper, were trying to make neurons of desired subtypes was to add factors on to his basic core factors. And so a number of people succeeded in doing this, and this can get complicated, but what happens with these neurons is oftentimes you get a lot of mixed populations, different types. And so what my lab decided to do is take inspiration from Coco Chanel, who says, before you leave the house, look in the mirror and take one thing off, to simplify the whole problem. So these guys, obviously fashionistas, Kevin Eden and Joel Blanchard, came up with an idea to try to make a useful kind of cell type. And I'm just going to, it's been published a few years ago, but I'm going to take you through it just so you can see how good these neurons really are. And then I'll tell you how we make a lot more. So peripheral sensory neurons are really important neurons, and we know a lot about their development. They detect pain, itch, and touch. They come in 3 flavors, track A, B, and C, that are different sizes and have different functions, and the Cs in particular are affected by neurodegenerative disease. So people have been studying these extensively, and Joel and Kevin noticed that family members of the Wernicke factors, neurogenin 1 and 2 and BRIN3A, looked like ASCL1 and BRIN2. And they thought maybe we could just take those and put them in fibroblasts and see what happens. So they did this experiment. They put in either neurogenin 1 or neurogenin 2 and BRIN3A, and if you put them in alone, nothing happens. But this is a video, and you can see that a cell here will start to change from a fibroblast. These are expressing a synapse in TD tomato and stretch out axonal-like processes. All my pictures died last night on the plane, so if everything was weird, I'm sorry. But you get plates full of these neurons. You can do it in human and mouse, and there's just a few things I want you to notice. The neurons are good. They fire spontaneous action potentials, and they have specific currents that are only found in these peripheral sensory neurons. What I thought was interesting is that when you use the Wernicke factors, you get these complicated neurons. When you use our factors, you get these simple neurons. That's exactly what the neurons look like, the exact shape of the neurons in the animal, and we get almost entirely those neurons. Second thing is, and this is interesting to me, when we looked at them, we were expecting neurogenin 1 and 2 to make different proportions of these neurons, as had been seen in the mouse, but instead we always get the same proportion of all 3 subtypes, and the soma size actually is co-reprogrammed with the expression of this marker. So the cells are receiving a pulse of these 2 factors for about 8 days. We let them mature for 5 or 6 days, and they resolve into these equal numbers of 3 subtypes of neurons. And we know that these are, we've later done RNA-seq on these. I don't know if you can see it, but their RNA-seq bulk profiles overlap almost entirely. There's only 5 or 6 genes different between them. And so if you want to use these kinds of neurons, you can actually do calcium imaging, if you can. For example, that should start going. And you can see selective responses even to these different ligands that mimic heat and pain, itch, and noxious sensation. And so you're reprogramming these cells with the same input, but you're getting diversity, but just some diversity of the neurons in your output. And it's very, very reproducible in mouse and human. So with these and other neurons, I'll tell you about, we think we can take fibroblasts or blood from patients, we can convert them into the neuron subtype we like, we can do that at high efficiency, and we could look at things like drug responses. And then we could also look at different individuals, different genetic backgrounds. We can also convert these into iPS cells, and the iPS cells plus transcription factors can also be used to make these neurons. So it's ready for systems biology people to tell me, can we do something with these neurons? So the model that we have been thinking about is a little different now than the traditional developmental Waddington lineage model where you have to fall down a pathway. And we're thinking reprogramming is maybe a little more like roulette. You mess up the cell, you tell it don't be a fibroblast, you give it a little information, and it can only fall into a couple of holes, and it's not turning into different things. So what about the rest of the brain? Can we actually start to make cells that are either a subtype of interest or that express genes that we know are of interest and maybe networks related to these complicated diseases like schizophrenia, autism, and anxiety? So we could have two possibilities. I could hire 2 postdocs for every type of neuron in the brain at 20,000 people times 6 years, or we could try to figure out, is there some sort of code? So Rachel Tsunemoto, a graduate student in the lab from UCSD's neuroscience program, bit the bullet in her 4th year and said, my other project failed, I'm going to do it. And she cloned every POW and BHLH and a few other transcription factors that had been reportedly found in neural lineages and some that had not. She did a giant screen of these 600 pairwise combinations, screening for a number of neuronal markers in triplicate. And then she came to my office with her results. And of course, I was really worried that it would be 2, and no students would ever come to my lab again. But instead, it was actually 76 out of 600, which we actually think is an underestimate. So we found 76 new pairwise codes that transient expression can convert these fibroblasts into things that look very much like neurons. Cajal would recognize these shapes. Interestingly, we used OCT4 as a negative control, and it actually works really well. So you can't always predict what's going to work just by looking at the end state of the neuron, something we can talk about. So why do I call these induced neurons? You know, they're not exactly like the normal neurons, but all of them fire beautiful action potentials. And remarkably, they also show signs of wiring together in the dish, which normally for our iPS-derived neurons requires co-culture with glia. So this is just, 14 to 21 days from when they used to be a fibroblast. So this is advanced neuro stuff. So to find out what they really were, what we did is we decided to take 8 different populations of endogenous neurons that we had labeled with different CREs, and with the same tau GFP marker that we made the induced neurons, we remade 35 of these in duplicate, and we did RNA-seq of all of them. So that took quite a while, but it was very informative, I think. So what we can see looking at the bulk expression profiles is the induced neurons are green, the endogenous neurons are purple, and the fibroblasts are over here. So they're definitely closer to the endogenous neurons, and in different ways you look at them, they mix in. The genes that are differentially expressed from MEFs are all very beautiful neuronal genes. We then compared the endogenous and induced neurons of their genes that were different from MEFs and intersected that. And so we called that a core neuronal transcriptome. That is what your developmental biology process can build in your brain that matches that I can build in a dish with just 2 transcription factors. And we found a good number of 6,000 genes that actually represent this transcriptome, and those are in orange, and they're the greatest hits of neurobiology. We also, from this, could ask what's wrong in the induced neurons. Those are the greatest hits. What we found mostly were leftover MEF genes, Hox genes, and some of those we show later are actually just not fully reprogrammed, but some are actually because we have contaminating MEFs coming along when we sort. So these are actually better than they look. The missing genes found in the brain and not in our induced neurons were mostly glial genes. So we actually think we got a very good representation of the transcriptome of a neuron in this experiment. And you can see the percentage of the matched genes in all the induced neural populations and the endogenous populations here is pretty similar. Some are worse than others. We can do a lot with this data set. One thing we tried to do is work backwards from the core. We could use Homer and Ingenuity pathway to look for motifs, and we found a few things from this. One thing we found is we found a lot of things that were on in the MEFs and off in all the other neurons, which we think are repressors. So the other group are activators, and to make a long story short, it's in our paper, which was just published in April in Nature. We find a lot of shared candidate repressors, but we find that the activators are more diverse. And so this could represent a model where when you reprogram, you get rid of the genes that are turning off neuron genes, and then when you want to make your diversity different activators come on to diversify, but we need to do a lot more work to show that's true. But the system's very powerful because we can take the candidate repressors and we can knock them down very easily in the dish, and we can see, for example, if we knock down REST, it improves reprogramming. So we're really just scratching the surface of this data set. The next big question is, I told you we found a core that's common to all of these neurons. Are the induced neural populations different from one another? And so to look at that, we did a lot of things. One, we looked for unique genes, and the dark genes are those that are unique to each population. And really, there are very few genes that are unique to an individual population of neurons. In fact, when you look in the brain, you find a gene that's special for your population in a region, but then you'll find it's in other neurons in other regions. So neurons don't have a lot of their own genes. They seem to make diversity using combinatorial coding. We also looked using WGCNA to see if we could find networks that might represent cell subtypes. And we did not, but we did find networks that were, as expected, related to the sets of reprogramming factors, BHLH factors, POW factors, but we found a lot of these, which I think are very interesting, synergistic networks that seem to be co-regulating the genes. However, I learned that WGCNA only lets a gene be in one module at a time, and that's not how the brain works. So we've been working on maybe finding more neurospecific ways of organizing these genes into networks, and I'm very interested in talking to people about that. If you're a neuroscientist, what you care is what kind of neuron it is, and that's its neurotransmitter. And we found evidence for different neurotransmitter expression in different populations. We tried to map them to cell types, and what we can find is the core maps to most neurons, the induced sensory neurons map to few, and then each individual cell can kind of map to individual reprogramming factor pair, make cells that map to certain kinds of neurons. But again, this is with a small number of genes, and we really need better transcriptome to neural subtype maps for the brain. Finally, we did single-cell RNA-seq, and we showed that when you make induced neuron groups and look at single cells with each factor, they cluster together. So you're getting a common output. They're not mixing together, except for in cases where we have extremely low reads. They all express core genes. When we try to subcluster them in groups, they don't seem to subcluster very well. And then when you look at the unique genes, you get very interesting things. The induced sensory neurons I showed you before express piezo2, which is a mechanosensory gene that's selective for those neurons, and others express, for example, nicotinic receptors that I'll show you a picture of. This is a human GWAS hit for nicotine addiction. So how much time do I have? Like, am I done? 5 minutes? Or it's okay. I'll just show you. This is a movie, because I just have like two more, basically, little things. This is a movie of human iPS cells that we genetically engineered two factors into and induced, and you can see them turning into neurons, and Marius Wernig and Tom Suda have done this with Neurogenin 2 as well. But you can see them over the course of about 7 days just rapidly changing their morphology and moving into neurons. And in this case, we get what looks like almost 100% conversion into these cells. We still don't know how their subtype compares to the fibroblast subtype, but I'll just skip to this picture where you can see this change over time, and we can get very high yield conversion with this tool. So we can make, we've tested now something like 30 different factor pairs in human. They all work. We're working on figuring out how they might be different or the same from the mouse. Can we use these for psychiatric disorders? Well, autism is actually an interesting disorder. What we noticed in our induced neurons is some have this thing called voltage sag that's mediated by this HCN1 channel, and some of our induced neural populations show that, and others do not. This is an inhibitor of it. And there was a paper from Tom Sudoff and Marius Wernig showing that voltage sag actually may be affected downstream of certain autism mutations. So if you wanted to study this, you would like to do it in cells that have this property. And there are a lot of examples of that. Finally, nicotine addiction, there's a cluster, a haplotype of nicotinic receptors that's a strong, strong hit for increased nicotine addiction. And we wondered if we could make these neurons, because it would be nice to compare the people with and without this set of SNPs. And we can indeed use our screen to type in these genes ask which induced neuron transcription factors will make cells that do that, and we can make cells that fire and are blocked selectively by the nicotine receptor antagonist mecalamine that we were expecting, and we showed some more data of that, like that in our paper. So in summary, I've told you that I think with reprogramming, we can turn fibroblasts into things that look a lot like neurons. They share a core with all neurons of the brain. They also have diversity that may be relevant for modeling disease. And of course, someday we'd like to know whether we could also mimic these kinds of things, the extrinsic factors such as aging or the role of experience and connectivity using transcription factors. I don't know if we can or cannot. So the Feynman quote, what we can't create, we cannot understand, may apply here. And I want to say that we've put our transcription factor input transcriptome outputs together on BioGPS, so you can go in and type in any gene and see if I have some ways to make cells that express that gene. We'd like to make this better to work with networks, and we hope that these cell autonomous features of neuronal identity and diversity that we're uncovering will help to inform our understanding of the RNA-seq studies that people are doing of all different animal brains. So that's my lab and my funding. Thank you all for your attention.
So there's probably time for one quick question. It could be from Trey. Okay, it's going to be from Trey.
Yeah, thanks for a great talk, Kristin. So you have all these exciting TF combinations that can induce neurons and subtypes. I think this is a correct question. How many of those do you think are biological? Meaning, are those the pairs, or are those pairs at least part of TF sets that are actually used during development to create those neurons? One could imagine that there could be many ways to, many roads to Rome. Biology chose one of many roads, or is it that there's pretty much like 7 roads to Rome and biology uses all 7 all the time?
We definitely find factors that are not predicted by the end state of the neuron that work. But what we have not yet done, and people are starting to do, if we did start with the end state, would that also work? In our IPS studies, we've been able to show, for example, using antibody screens, that we can actually reprogram cells to IPS cells by intersecting this transcriptional network, inducing it at different points. And others have shown that as well. So this idea of reprogramming is if you destabilize cell type A network, and then you impart cell type B network, you may be able to trigger that network, which is self-perpetuating, in multiple different ways. And so our ways for these happen to involve transcription factors that go on and then go back off. And if you track the endogenous ones, some will go on and off, and others will go on and stay on. So I'm very interested in how to figure this out.
That's great. So all the speakers are going to be around at lunch, but we're going to get the next one up, and you're going to plug in, is that right? Okay. You're the only thing standing between us and lunch.
Oh, yes.